Enzyme catalysis. In order to understand the concept of enzyme catalysis, we should understand what is enzymes. So, enzymes are proteins that act as a biological catalyst. Sometimes it is also called as biocatalyst or biochemical catalyst. Basically, catalyst will accelerate the chemical reactions. So, the molecules upon which enzymes may act are called as substrates, and the enzyme converts the substrate into different molecule. That is what we get, or it is known as products. So, they will catalyze many reactions which will occur in the bodies of animals and plants to maintain the life process. So now we should know what is enzyme catalysis. Enzyme catalysis is a procedure in order to increase the rate of virtually all the chemical reactions within cells by the active site of a protein. So that is what we call it as enzyme catalysis. Hence, most enzymes are proteins and most such processes are chemical reactions. So, catalysis of biochemical reaction in the cell is vital because many but not all metabolically essential reactions have very low rates when they are uncatalyzed. So, we say that enzyme catalysis is a procedure in order to increase the rate of virtually all the chemical reaction within the cells by the active site of proteins. Hence, enzyme catalysis follows a mechanism. Enzyme catalysis reaction will usually take place or it will proceed in two steps. First, it is saying that binding of enzyme to substrate will form an activated complex, and then decomposition of the activated complex to form the product. So here, if we consider E to be the enzyme and S to be the substrate, this will bind and it will give you the activated complex, that is your ES. And second, decomposition of the activated complex will lead to the formation of the product. So, this is the basic mechanism of the enzyme catalysis. Hence, enzyme catalysis has certain characteristics, that is high efficiency. We can say that one molecule of enzyme may transform 1 million molecules of reactant per minute. Hence, enzyme catalysis is having high efficiency. Second, it is highly specific in nature. That is, each enzyme catalyst cannot catalyze more than one reaction. For a specific reaction, we use specific catalyst. Then, optimum temperature. Enzyme catalyst gives higher yield at optimum temperature, that is considered to be 298 to 310 Kelvin. So, human body temperature, that is at being 310 Kelvin, is suitable for enzyme catalyzed reactions. Then, it maintains optimum pH, that is the rate of an enzyme catalyzed reaction is maximum at the optimum pH range, which is from 5 to 7. And next is activators. Activators like ions, we could consider it as sodium ion, calcium ion, manganese ion. They all help in the activation of enzyme which cannot act on their own strength. Then coenzyme. Coenzymes are the substance having nature similar to the enzyme and their presence will only increase the enzyme activity. So, mostly vitamins act as coenzymes. And what are all the effect of inhibitors? Inhibitors generally slow down the rate of enzymatic reactions, but the use of many drugs is based on enzyme inhibition action of those drugs in the body, because inhibitors are also important for certain drugs. So these are all the specific characteristic of the enzyme catalysis. Hence, few examples for enzyme catalyzed reactions. So the first one is inversion of cane sugar. Here, invertase enzymes will convert cane sugar into glucose and fructose. Second is conversion of glucose into ethyl alcohol. That is, zymase enzymes will convert glucose into ethyl alcohol and carbon dioxide. Third example we could take is conversion of starch into maltose, and so diastase enzymes will convert starch to maltose. So these are all the general enzymes catalyzed reaction. How do we use catalyst in the industry? The most important application of catalyst in the industry we do is in the process of Haber's. That is, Haber's process is used to manufacture ammonia. For example, here if we take N2 plus H2 is going to give us ammonia. So the catalyst used in this reaction is iron with a suitable promoters like calcium oxide, silicon oxide, and many more. The second application of catalyst in industry is Ostwald process to manufacture nitric acid. That is, in this, a mixture of dry air and dry ammonia in the ratio of 10:1 by volume is compressed and it is passed into a platinum gauge, which is the actual catalyst being used in this reaction. And this reaction takes place at 800 degrees Celsius. The last application we could say as contact process, which is used to manufacture sulfuric acid. Actually, in this, sulfur trioxide is being formed when sulfur dioxide will react with oxygen in the ratio 1:1 at a temperature of 400 degrees Celsius to 450 degrees Celsius and a pressure of 1 to 2 atm. But the most important thing to note is vanadium pentaoxide is acting as a catalyst for this reaction. That is, vanadium pentaoxide is being used in contact process in order to manufacture sulfuric acid. So from this we could conclude that catalyst is playing an important role in our body as well as in the industry. It has its own importance in our day-to-day life.